Okay, I hope you're going to cut off this. So I'm sorry about the confusion, but that's my first webinar with GoToMeetings, and it's a bit more complicated than MS Teams, which is itself complicated. But anyway, who we are. I started as an ImageJ developer in 2003. Actually, there was no such thing as ImageJ development then, but I had to solve some issues related to the analysis, image analysis of data in my PhD, and we didn't have funds to buy more expensive image processing software, and this is how I started. So after my PhD, I moved in a slightly different direction and joined IMEC in 2008. But I still went on with my development, let's say, activities, although in a more high level. And since 2021, I'm also part-time adjunct professor at the Bulgarian Academy of Sciences. Then we come to my colleague Sumit, who started his master thesis with me back in 2015, I believe. And then he joined the institute in Germany in 2018. Where he's pursuing now a PhD. Sumit is the most active developer in the active segmentation toolbox, and we are going to demonstrate the software as well as introduce some topics of image analysis. So what's important about image analysis in general? There are two typical tasks. First of all, a segmentation task, let's say. In that we ask typically how large an object is. And secondly, a classification task in the sense that if this is a member of X. So the image analysis is more an art than a science in the sense that, I mean, besides the classification and segmentation, a lot of things can happen under the hood. For a lot of tasks, we demand a lot of signal processing and also machine learning, as it will become apparent in this presentation. Okay, so what is, first of all, the traditional way of doing an image segmentation? If you see here on the right, there is my favorite blob image in ImageJ collection, which you can segment by simply thresholding it. And in that case, it's rather easy. So any of the automated methods works well for that image. But in general, things are more complicated when it comes down to microscopic images. So we have to find a threshold, and there's some automation in that, but it doesn't work in all circumstances or in all kinds of images. So after the segmentation, after this thresholding step, we can binarize it, and then we see something like that, what you see here. Okay, I don't know where they're two, but anyway, both are relevant. So then you can also do something like watershed to cut the large objects in pieces in case sometimes this is pretty successful, in other cases it isn't. And then finally you can get your regions of interest and measure them. So this is a traditional approach. It works well for some images where the cells are not really touching, or there is slight variation in the image contrast, as you have seen here in the example above. but in other cases we have problems, and this is exactly the reason why we started developing the active segmentation. So at that point, I'd like to introduce you to the concept of a feature space. So what is this space? This is a representation of the original image where certain features are enhanced and others are suppressed. For example, we can use a generic feature sets such as the Gaussian or the gradient or the Hessian or curvature that actually decompose the image along some mathematically formal way. And out of such components, we can build other features that are sensitive to objects in the image, such as blobs, for example, or lines, or tubes, or corners. Now, as an example here of the primary feature space, I have used the one default image from the collection of ImageJ, that's the fly brain. So here in this image, if we convolve it with a gradient, so in the what you see here in the filter in the X direction, you get something like a shaded view. And if the direction is vertical, the shade goes in a different way. So goes vertically. So these are the two primary components of the gradient in the X direction and then Y direction. And if we proceed further along the decomposition step, we can get some more features that look in not really revealing way, but are useful in automated approaches. So you see them here. These are the so-called components of the Hessian. So out of those, we can compute more useful features, which we call geometrical features. For example, here again, using the components of the gradient. So that's an image of cells. We can compute the isophots, so that means the levels of the image that are at the same contrast level, or we can compute the directions of the overall gradient and its magnitude. And if we use the second order features, we can compute something called Laplacian and the eigenvectors of the image that point in the direction of increasing curvatures. So all that being said, it's still a more of an art than science, because then we have to find a scale. Because in order to suppress the noise, we have to convolve the image with a smoothing filter. And that smoothing filter can have a different size and therefore can suppress different sizes of features. Now, what we can also do is to use some decompositions of these filters. And then again, you see in an example that if we, for example, use an isotropic filter, a Laplacian, we can get one type of segmentation, so that corresponds to a cell, to the nucleus of a cell. But actually, we see that the nucleus has a more complicated size and morphology. And if you use the composition of the same filter, we can get both lobes of the nucleus quite well. So as I said, I mean, the trick is actually to select a scale, and we can also do it in automated way by using certain observations. And the observations are that if you look at the natural objects, such as here, so these are mice grinds, for example, they have only limited size, and their structures inside the grains are also of limited size. So I mean, if we imagine something like a scale space, the objects are represented in a limited way. So there is certain peak where the scales of the objects are positioned, while the noise itself has features of all scales because it's random and it can change the contrast in an uncorrelated way. So we can use that if we systematically apply the filters also along this line, so to increase the scale of the image of the filter so that we can sieve off more and more features. But in order to recompose the image later on for the segmentation task, we need some machine learning that can identify, first of all, what are the relevant features of interest, to combine them, and also to focus on the scales that are also relevant. And then now we come to the platform. How do we combine all this theoretically into a practical segmentation platform? So we combine three domains here. The first one is signal processing, the second is machine learning, but we also need a domain knowledge of the researcher who needs to perform the segmentation task. So the researcher gives off samples that are relevant for the task, and the platform itself learns the features and the segmentation. So the active segmentation is written in Java, and it's based on the standard ImageJ. I mean, we use it as input-output and visualization functionality, and we developed it since 2015. You can download the platform from this address here. It's the development site is GitHub, sorry. And now I'm going to guide you through the platform itself. What you see here on the left is the main screen, where we have the main menu of filters, feature extraction, evaluation, and learning. And this is exactly the way how you should work with the platform. So we start from selection of certain filters, let's say, and then we go on towards annotation and labeling of the image. Then we select the learning algorithm, and then we evaluate the results. So in the first place, how do you install the software? It's actually an ImageJ plugin. So if you're versed in ImageJ, you don't need additional explanations, but I assume that you're not. So we have compiled an installer for Windows users. So it's a binary that does not require special privileges. So you can install it almost in any kind of organization. And then there are archives for Mac and Linux users that we will update shortly. Basically, for Linux, you have to just unpack the archive and configure ImageJ to recognize the plugin directory. While for Mac, I'm not really familiar because I've never used one, but Sumit can give more information in case you're interested. So what you see here is how the GitHub repository looks like. And then if you click on the releases, you can download both the Windows installer and the jar archives for the complete release. Now, once you install the platform, you can find the platform under the plugin segmentation, active segmentation. So once you click active segmentation, you will see this screen, you'll see this frame below, where you can either create a new project or open a project. And then how can you use your data, the plugin with your data? On the first place, we advise you that you create a project. What is a project? It's a folder tree, what you see here on the right, where we have different images that are computed by the platform. And we have different stacks that are copied in the project. And we also have a metadata file which describes which features are used, what filters need to be activated, and what their settings are. So the first thing you do is to create a project. Once you create the project, you can go on to the next step. And the next step is to open this screen that I have showed before. And this is the sequence of working. You select the filters, then you select regions of interest, then you choose what kind of machine learning algorithm you need to run. And then after the algorithm segments the image, you inspect the result, and possibly you decide to refine the selection. We start with the selecting of the filters in the menu. So you will see another frame here that says filter, and in the tabs of the frame you will see all the filters that are installed. In an empty project, by default, all the filters in the platform are loaded, but you can exclude some of them, and I'll show you how. What is also important here are the defaults, which resets the parameters in case that you're not satisfied with the results, and the parameter window here, where you can type in the values of different parameters that are related to the filter of interest. When you click next, you navigate to the next filter tab, and you repeat the same procedure. You can also decide to disable a filter, and for that you click here on that enabled button. Or you can also decide to include the filter to enable it back, and you can do that by clicking here in the window. Finally, what you see here, the picture, that's typically the response in the spatial domain of the filter. Sometimes it's the response in on the frequency domain, and that's basically a developer choice. Whatever to show you here in that screen. Now, if you want the filter to be computed, you press compute, and compute actually computes all the filters. It also checks if they were up to date. So in case you change the parameters, it can recompute filters. And what is also interesting is that you can inspect the filters, which I'm going to show you next, and you can also save the configuration in the project file. Now, if you want to inspect the features, this is how you do it. This is the filter visualization, where you can see also the regions of interest that have been marked as samples of the objects, and you can also navigate both in terms of slices in an image stack, or in terms of features in the feature space. So once you click next, you load the next filter, actually, the next filter response, and you see here the name of the filter image. And you can check how it looks like also by clicking view, so that undocks it, undocks this filter response, and you can also inspect it from the ImageJ main window, so that you can run histograms, you can threshold it, anything you want basically, but that's for more advanced use. Right, and then what you also have to do is, of course, present examples. And how do you do that? Here in that part, you define how many classes you want. Our advice is 2-3 classes, let's say. I mean, if it's a simple segmentation task. Sometimes it's more complicated if you have really many kinds of objects in the image. And then what you do after you configure here, so you give the names of the classes and their colors. What you do, you select some ROI instrument in the ImageJ main window and start presenting the ROIs to the platform. So double clicking. So first of all, clicking on the button here adds it to the ROI list here, and double clicking on the ROI list here on the item in the ROI list actually removes it. And it's up to you. You can also zoom in here in this window and really draw your feature, your samples accurately, or you can just present, like in our case, big squares or rectangles with the hope that the platform is intelligent enough to learn what we want it to learn without being more specific. And that's actually one of the problems in the field, that we're dealing only with partial ground truth in all cases, because even if you're, as an expert, have the morphological knowledge of what you want to segment, sometimes the image is simply not clear enough so that you cannot really segment out the entire object you want. But we give you the possibility to actually mark only the part that you're certain about. Right, and then in the learning configuration, there are predefined algorithms. So you can just select something like, for example, random forest or SVM. SVM starts for support vector machines, but Sumit will present that in more detail later. And once you learn the examples, you can invoke the masks here, and you see the results, the outcome for yourself. This is the Freya project, which I have also included in GitHub, so it can be downloaded and checked for yourself. Applications to plant science. Here, basically, we have selected something that may be of interest to you. For example, there are two the common segmentation tasks here. One is about object localization, where you want also to have some kind of dense segmentation and really segment the object of here. For example, here are seedlings, so you want to segment the seedlings and measure how long they are. Or it can be multiple object segmentation, and then you can also check other things. These are the data sets that we have selected, and we will demonstrate further. One comes from the paper of Nucleus J, second edition, which is the right data set. These are nuclear data, and the other are RGB images of seedling coming from this paper here, Dobos. So that's a task in the plant phenotyping where the hypocotyl length is determined in order to classify something. Okay, now we come to the machine learning part. So maybe Sumit, you would like to take over here.
Okay, let me just give you the power here, Sumit.
Yeah.
Okay, you're presenting now.
Can you see my screen now?
Yeah, all looks good.
Yes.
Okay, so let me. Okay, so as I'm going to show you several examples from the plant research, or I will say the plant science, and one would like to segment this. I will say this nuclei or this hypocortical. Then they want to determine the length of this hypocortical because it's very important for plant phenotyping. Okay, and there are several ways to do segments such a data sets. One is thresholding, as you see that even in the Nucleus J, they use the thresholding way out to segment these data set. And the other way is with the machine learning based tools. And our toolbox is, or our platform is based on this artificial intelligence based algorithms. Okay, so there are two ways to the way we can define the machine learning. So one is the traditional classifiers for segmentation, which are very simple and require less training for data, require less training data, and they even don't require the dense labeling. When I will explain in detail what do I mean by dense labeling, and they also require less time to train. On the other hand, these days, you know that a lot of people are going towards the deep learning based segmentation tools. And here, these deep learning based tools are much more complex to understand, and they require dense labeling. If I talk about dense labeling here, it means that you need to label each pixel here, whether it's a class 1A or a class B for segmentation. And then this deep learning based model require longer time to train. Although the accuracy of these tools are much more higher, I will say that deep learning based segmentation tool is much more higher than the traditional classifiers. But there are advantages or disadvantages of both the techniques. So in case of the traditional classifiers, one can do the training much more faster rate, and then it's very in the starting of the algorithm, it's very difficult to get dense labels because we are not sure about whether it's a part of object A or object B. And as a state of art, now people are combining both. So in order to get some of the samples where the users are sure about the data, they perform the traditional classifiers, get the dense labeling for them, and then they give the input to the deep learning based classifier to get the higher accuracy. And in our platform, in actual, we support both. We have the traditional classifier as well as the deep learning, but in our current release, only we expose traditional classifier for the community. We are still working in improving this deep learning section. Okay, then how we do the segmentation. First, we will input these images, and then we extract several features at several scales. Here you can see that because it's more like a tube, so you will find that we extracted the tubular structure at several scales, as well as the texture information. I will say a lot of complicated features. And once that's done, then expert will provide us some samples for training. And how expert can do that? It can select a square ROI or a very specific region of the hip. Like here, you can see that I marked a very specific region of the hypocortical. And the good thing is you don't need to mark the whole region. You can mark only the portion where you are certain and where you can mark it very easily. And then the third class, we defined it here as a non-hypocortical portion, and we labeled some samples from there. Once you are done with very few samples, labeling a few samples, we do the feature selection step. As we know that we extracted a lot of features, and it can happen that it will, if we use all these features, the training time will be very high, and then all the features are not that informative. So based on the heuristics, we select certain features and reject certain some of the features. And then the next thing is about the normalization of these features, because these are at different scales. One should normalize between in the same range, so that one feature should not get higher preference over the other during the learning, because these classifiers are more sensitive, are very sensitive toward these features, if to the one feature, if they are in different scale range. Okay, once after that we paint the classifier, and once when we get the mask out of it, and if we will overlay the mask with the data set, you will see something like this. And here you can see that where the algorithm is performing wrong, not well. Like for example, here you can see that it's over segmentation, and then the expert can mark the areas where the algorithm is not performing well for the next iteration. So then the expert will provide some few more samples in several slices in the stack, and then the next iteration will start, and this step will keep on going. And on a similar way, I would like to explain this another data set where we are interested in the nuclei segmentation as well as the chromocenter segmentation. So okay, we input one image stack, and then we extracted several features like blobs. Here you can see that we have the blobs, and then we have the even the textures, and then we start with labeling very few samples. Here this is a background area. Here we said that it's okay, it's a part of the nuclei. We don't need to even label the whole nuclei. And the third, we said that okay, these are the chromocenters, and then on a similar way like previous, we reject some features and we normalize those features. And then here, once the training is done, you will get such a mask, and when we overlay such a mask over the actual training image, you will find where the algorithm is performing right, or where it's being wrong. Here you can find that okay, algorithm is not able to learn this portion. At least add this as a sample for the next iteration, and this process will keep on repeating till the researcher will say that okay, it's fine, the results are now perfect, and they can stop the segmentation at that point. With this example, I would like to thank for your attention, and I would also like to thank the interns that worked in this project from a long period of time with us, like Thomas, Mac, and I also worked in this project as an intern in 2015, and then Victor, Sanjay, and Joanna and Raghavendra. And I would also like to thank the various funding organizers to develop this platform, Google Summer of Code, INCF, and FWO. And I would also like to thank the In-Depth Academy for organizing such a seminar. And we are happy to take questions now.
Great, thank you very much, Samit. Thank you very much, Dimitar. It's very interesting stuff. So I'd encourage anyone who would like to ask a question to please write them into the question box. I know there's quite a lot to take in, and before you ask anything, let me ask first of all, is there any limit on the quality of the images that you need to perform this successfully?
Okay, so I would suggest that we generally prefer the TIFF stacks because you know that generally TIFF stacks are of high quality, and then okay, if the images are of really poor quality, then the algorithm will not able to do really well. But otherwise, we tried even on low quality images, the algorithm performed quite well. So there is no hard fast rule, but we recommend that if you really want a very high quality segmentation, it would be recommended that you should have at least some quality of data set. Not like we don't expect very high quality, but at least some up to a certain extent.
Absolutely. Okay, so if I know it's challenging, everyone's not most people aren't English first language, so it's even more difficult to write a question in foreign language. So can we proceed with the demonstration that you have, and then we'll come back and see if people have questions afterwards.
Oh yeah, sure.
Okay.
Okay, so I already installed this image version, and let me go. Okay, so here we have images started, and then you can see that in the segmentation section we have the active segmentation, and I clicked on the active segmentation, and now I'm going to open the project which is already in my PC. So here now we are opening a project. Okay, in a presentation here, I'm going to demonstrate the nuclear segmentation. Okay. Once the project is loaded, you will see this screen, and here first user can select or unselect the feed. Okay, here you can see that I am disabling some of the feed filters. And now we have these 4 filters, and it's up to the user. If they want to change parameter, they can change it and then press the compute button. And now you can see that it's computing because it's reflecting in some other color. Okay, now you can see that it's computed, features are already computed, and next you can, if you would like to visualize this, you can visualize them. Because the size of the image is very small, so that's why it's displaying it in a very small window. And next step is you would like to provide some samples for training. In order to do that, let me improve the quality of the image. Here, as you see that I already provided some of the samples for training, and one can label more sample for training, like there, create a bounding block and just press to the class where you want to add this label, and then you can do it for subsequent slices where you are sure about, okay, this is the chromocenter, and this is the nuclei, and this is the background, and you can keep on doing that. And then once you are done with that, you can save this ROIs or into the meta file. Once it's done, and then you can you should click on the train button. Now the training is started, and one can see that okay, so once the training is done, the overlaid image will automatically come, and here you can see the results of the segmentation, and you can go through the several slides. And if one is only interested in the masks, and one can get these. Okay, and then from now onward, like we know that where the algorithm is performing wrong, like here we know, okay, this is the part of the this is not correct, I want to add it here, and then even might be here. Okay, and then one can again click on training. Okay, when training is done, you can see that in the next iteration it's approved. And this step can keep on going till the expert is satisfied with the result. And as you see that it's very quick, and I labeled only very few samples in the whole stack. Okay, do you have any questions? And one can also go through like, so all this data set is stored in this folder. So here you can see that we have the images that are stored, and then we have the filter output for each of this stack slice, and then here we are storing the labels. These are the three set of labels, and they are archived. They are in archived format, and then these are the masks related to the data sets. And here in this folder you can find all the metadata related to the segmentation. And if I open this file, here you will find that we have all the ROIs list, in which class it belongs to, which zip file it has, which color it belongs to, as well as all the information about the project will be stored here. Basic information to, I will say that's from my side.
So let me ask, so once you have all this segmentation data, then what format does that come out in? And then can that be put into a, to do measurements and things like that? So what sort of format does it come out?
Can you repeat the question?
So the images that you get, or the segmentation that occurs, you get the segmented images. What output format do they come in?
Okay, so it's in the TIFF format. Output is in the TIFF format.
Okay.
But when we are planning to do it like to include several formats for input and output.
Okay.
But it's not done yet. Right now, everything like your output would also be in the TIFF format.
Okay. So that can then be taken away and done whatever people want to do with those
yes,
images.
exactly, because most of the so you know that whoever do the segmentation, they like to prefer the TIFF stacks to work around with, because TIFF stacks are generally of high quality. So most of the user prefer TIFF stacks. So that's why we started with the TIFF, and then we will keep on providing the other formats sooner or later.
Okay, great. So have you done much collaboration with any plant scientists yet? So Dimitar, have you had much collaboration within the INDEP, for example?
Not yet. So we are. I have mainly collaboration with the neuroscientist. We tried several data sets from the, I will say, the zebrafish domain or from the mouse domain, but not from the plant. I will say the plant science. But here I demonstrated you the two examples, because and it's really, you can see that so within so I used only very few stacks, but even on that stacks you can see the result, auto segmentation result. It's quite good. And if somebody is really interested, we can try on their data set and they can even try their own. If they have found some issues, we can help them to fix it, and we can say that it's worth to try toolbox.
Mm-hmm. Okay. Oh, sorry, Dimitar. Yeah, Dimitar, I muted you a while ago. Sorry, Dimitar. Let's have a look. Yeah, go on. Sorry if you got a comment. No. Oh, you're still. You need to switch your audio on now, I think.
Okay. So first one remark about the image formats. The JPEG images are not good, even if they are kind of easy to get from phones and cameras, because the compression introduces artifacts, which for the human eye look okay in terms of quality, but our algorithms are extremely sensitive to the artifacts that the JPEG compression does. So my advice here is use TIFF images, PNG images, bitmap, simple, anything without lossy compression to get optimal results. And then in terms of plant scientists, we tried a collaboration in Szeged in 2018, but then the platform was not yet mature enough, let's say, for production use. So we didn't have examples other than that. Yeah, I also demonstrated it 2 years ago in Antwerp on the Pictures to Numbers school. There were some plant scientists there. I mean, one of the examples, the Freya image is some kind of plant. I don't know exactly what it is, but I use it always in demonstrations because it has a large nuclei that are easy to segment. And yeah, that's it from my side in terms of remarks. Yeah.
So we have a couple of questions now. So a question from Narendra, and yeah, it says, "Nice presentation, thanks." I see that the labeling is done using rectangular boxes. Is it possible to do precise labeling, for example, polynomial boxes instead of rectangular boxes?
Yes.
I can show you. So like here, you can see that I'm using the rectangular box. Just go to the freehand style and then go to the screen here, and then you can do the very precise labeling wherever you want to do. Like this. This is just an example. Like you can do it by your hand very precisely.
Okay.
So it's just I will say that I showed you a very quick demo that where you don't need to provide even the very precise labeling, like just even the rectangular boxes. Using the rectangular boxes, you can achieve a quite good results. But like if I talk about the other data set, if as the let me show you the data set. Like in this case, you need to provide at least some good high quality labeling in order to achieve the good results. So it's all depend upon the data. So if whether they required such a high quality, I will say labeling or not. But it's possible. You can use multiple options from the image. So there are several options. You can use rectangular, you can use oval, or you can use freehand style or polygon selections. All these options are possible.
I see. Okay, so we got also a question from Christoph. It says, can you compare active segmentation with other labeling tools like Lastic or Weka? I'm not familiar with those.
We are using Weka. And actually, it's very much comparable to the trainable segmentation. This was the inspiration for our tool, but let's say it's like 3 generations ahead of trainable segmentations in terms of functionality.
Okay, so I can compare it more. So trainable segmentation use filters from I will say from outside the community. So it's already predefined filters. They use it. We in our case we designed our own filters, and it was very difficult to include our own filters in the trainable weaker segmentation. And then we decided to start from that point to make a platform where we can include our own filters, and even the filters from trainable segmentation can be included there. And then if I compare it with Elastic, principle of trainable segmentation or Elastic or a weaker segmentation or our platform is the same. Principle is same, but we have the different set of filters which are not available in I will say Elastic also. So and if we like to compare the results, so it depends upon the data set. In some data sets, maybe trainable segmentation will work better. In some data set, our platform will work better, or the Elastic will work better. It all depends upon the features and with that's how the things work.
Okay. Okay. Cool. Okay.
Well,
So
the advantage, I would say one more thing. The advantage over Weka, well, there is no advantage over Weka itself because we are using it as a machine learning platform for our own platform. But the advantage over the trainable segmentation is that in our platform, you can incorporate third-party filters, which is not possible or not easy at all in the trainable segmentation. The advantage over Elastic is that you don't need to import your data again in ImageJ. So if you run, let's say, Python, I would say stay in Python, use Elastic because it's based on Python, C, whatever. So it's one ecosystem. On the other hand, if you're using Fiji or ImageJ, you have very similar in terms of performance platform that is within this ecosystem. So you don't need to go over complicated conversion, pipelines and imports and stuff.
Okay. Cool. All right. So we don't have any other questions, but I think this is a sort of technique where people need to go away and listen to what you've said and work through it themselves to figure out exactly how everything works. And hopefully we can give people the opportunity to do that now we have this recording. So let me say thanks very much to Dimitar, and thanks very much to Simi very much.